Let's talk about our chemistry of life notes. First, you need to make sure that you know and understand atoms and their interactions. You need to know what chemical reactions are. You need to know what protons, neutrons, and electrons are. That was in our basic chemistry, and you should know that for the test. If you have not done that grad point, you should probably flip through it so that you have a good idea of what that's about. We're going to first talk about water and its special properties and diffusion. Water is what we call a polar molecule, which means that they do not share their electrons equally. So it is a covalent bond because they do share their electrons, but they share them unequally. And because of that, they have a little bit of a charge to them. So what happens is it attracts other ions, attracts other polar molecules, and because it attracts other polar molecules, it attracts other water molecules. Because it is a polar molecule, it is bent in shape. It is not linear. We talked about and drew pictures of linear molecules, especially with carbon bases like our macromolecules. But water is bent like this because of its attraction. Water, when they are attracted to each other, when the water molecules are attracted to each other, they are attracted between the hydrogen and oxygen because one's a negative and one's a positive. That bond is called the hydrogen bond. This bond gives water the ability to go up thin tubes. We call that capillary action. That's how plants get their water. It also gives water surface tension properties. The surface tension properties allow for some tiny organisms to walk across water without falling through. That was how we were able to put the paper clips in the water. This picture here shows the difference of capillary action, how in the glass tube the water wants to crawl up that. That's because of that hydrogen bond, the molecule being polar. It also allows water to resist temperature change, and this is important. You need to know this for the test. It doesn't, it's going to boil, it's going to take longer to boil. It resists changing that temperature. So it has a high specific heat. That's the amount of energy that it takes to make that water hotter. Why is this important? It's important because it helps maintain steady environment when situations fluctuate, and it helps to maintain homeostasis in our cells themselves. Molecular motion, it's important that you understand that atoms and molecules never stop moving, that they're always moving. You need to know that the molecules that move in a constant motion, that's called the Brownian motion. Temperature is a measure of this motion. The quicker things move, the higher the temperature is. Diffusion. This is when we took our pure perfume and at the front of the room, we let it off and we waited for it to go to the back of the room and we timed it. It's the movement of particles from an area of high concentration to low concentration. There's no energy required, and it results in the Brownian motion. So the particles are going to just naturally move from their higher concentration of all the perfume in the bottle into the air. There are some factors that affect the rate of diffusion, and you do need to know these for the test. One is the concentration gradient. The greater the concentration gradient, the faster diffusion. Next is temperature. The higher the temperature, the quicker those molecules are going to move. And the pressure. And you increase the pressure, it increases the particle motion. How long does diffusion keep going? It goes until we have equal concentration everywhere. That doesn't mean the molecules stop. It means that the molecules have the equal amount on each side. We call this dynamic equilibrium. And that's what this looks like. You have the same amount coming in as you have going out. It's not like things stop, they just even out. So now we're going to talk about our life substances, and these are things like our macromolecules. And what you need to know and understand is that carbon is the background of that. And the reason that carbon is the background of that is that it has many electrons that are available for bonding. It has 4 bonds. So it's a good backbone or skeleton for bigger molecules. It bonds very easily with hydrogen, oxygen, and nitrogen. And you'll find as we start talking about our macromolecules, that is what they are usually made of. And the reason for that is because they bond easily with those. They bond with using covalent bonds they share, and they can either be single bonds where you have one pair of shared electrons, which is two, double bonds where you have two pairs of shared electrons, which means four are involved. At triple, you have three pairs of shared electrons, and that means that you have six involved. When carbon atoms bond with each other, they can either form straight chains, branch chains, or they can be ringed and shaped. They're very large, so that way they have these different shapes that will accommodate those. The isomers are compounds that have the same chemical formula, but you have a different 3D shape. And organic compounds that contain at least one carbon chain, so that the carbon comes off of it. So now let's talk about our macromolecules. This is the big part of the test. Macromolecules are made up of monomers. That's the single unit. They come together and they make polymers. And those are linked together by bonds, peptide bonds in some cases, and hydrogen bonds in other cases. And their monomers come together to make the polymers. The first carbohydrate we want to talk, the first macromolecule we want to talk about is carbohydrate. Carbo means carbon, hydrate means water. It's composed of carbon, hydrogen, and oxygen. You need to know that for the test. The monomer of carbohydrates is monosaccharides. You need to know that. Monosaccharides are simple sugars. They end in OSE, and glucose is an example of that. They are linked together, monosaccharides, to form carbohydrates. So that's the monomer. You need to know that. Disaccharides are carbohydrates that are made up of two of your monosaccharides, like sucrose and lactose. Polysaccharides are polymers composed of many monosaccharides. Examples of this is starch, and these are good for long-term energy storage, especially in plants. Animals can break down the starch that you get, so we eat the plants and then we break down that starch. But the long-term energy of these polysaccharides that's stored in plants is called starch. Another example of this is glycogens. This is our second example. This is what the long-term carbohydrate is stored in animals. We break down the glycogen to obtain glucose because we use glucose to make energy. Cellulose is also known as fiber, and it's a structural molecule that you find in cell walls of plants, which is really important. It keeps them straight up and down, and animals cannot break this down, so it just kind of goes right through our digestive system. Our next macromolecule we're going to talk about are lipids. And lipids are fats. They're used for energy storage, insulation. You have a protective covering on some organisms, some organelles, and on some cells. And it's a major component of cell membranes in our body, which is very, very important. Fats store energy, and they are energy storage molecules. They are made mostly of carbon and hydrogen, and you need to know that. You need to know that the monomer are triglycerides, which is a glycerol molecule and three fatty acids. That's the monomer of fats. That fats are what we call hydrophobic, which means they fear water, so they want to be away from water. They repel water, which is important in terms of our cell membranes. There are three different types of categories of fats that I want you to know. First is saturated fats, SS. They are solid at room temperature. You need to know that. They are single bonded. Next are unsaturated fats. These have double bonds in their carbon chain. You need to know that for the test. And they are liquid at room temperature. The third big category are our phospholipids, waxes, and steroids. That's our third big category. Our phospholipids is what makes up our cell membrane. And phospholipids are unique in that they have hydrophilic and hydrophobic parts. One side of it likes water and the other side doesn't. You have waxes, which is hydrophobic coatings composed of fatty acids and alcohol, and steroids, which is important in digestion and the making of male and female hormones. Our next macromolecule that we're going to talk about are proteins. Proteins are composed of carbon, hydrogen, oxygen, and nitrogen. Sometimes it has sulfur in it. There's a lot of functions of proteins, and proteins are very important. You need to know all these functions. First, they can be used structurally in terms of building things like hair, nails, and some animals' horns and hooves. They help in the movement, the contraction of muscles. They can be stored in different things like different foods, egg whites. They can be stored in red meat, that type of thing. They're very important in our immune system. They create antibodies and in the membranes. They help transport things like hemoglobin and other things across cell membranes. They signal things to start and stop in our body. They tell hormones when to be secreted. And more importantly, the most important thing is that they are catalysts or enzymes. The monomers of proteins are amino acids. Amino acids are put together by peptide bonds. These are covalent bonds between amino acids, but they are peptide bonds is the type. They're often called polypeptides. There are 20 amino acids. You need to know that there are 20, and you need to know that the monomer of proteins is amino acids. Here's an example of how complicated amino acids can get. And what's really important is proteins have so many different functions. They have also many different shapes, and their shapes are going to determine what their job is. Proteins need a particular shape to do their job. They can be very simple or primary, like a little chain. They can start folding up on each other or coiling up on each other, called secondary. You have tertiary, where we have some of those secondaries that are mixed amongst the primary, or you can have quaternary, where you have all of these that are mixed together. They are very, very complex molecules. Remember, we said one of the biggest functions is being enzymes, and they are a certain shape to be certain enzymes and to help regulate certain chemical reactions. Proteins regulate chemical reactions by changing the activation energy. That's really important. It speeds it up by helping to lower that activation energy. They can be affected by temperature, ionic conditions, and pH. All three of these things can change the shape of the protein. Changing the shape of the protein will change its job, so it can change how that enzyme can actually work. Please know that if the word ends in ASE, it's probably an enzyme, and that can be a good hint for you on your test. Next thing is nucleic acids. And we're going to talk a lot more about that in later chapters when we talk about DNA and RNA. But I want you to know that an important function of nucleic acids is that it contains the information to make proteins. And we already know how important proteins are. So nucleotides are our monomer, or our unit of our nucleic acids. It's made up of a phosphate group, a nitrogenous base, and a 5-carbon sugar. That's what a nucleic acid is made of. And those nucleic acids is what makes up your nucleotide. It's the building block. Your nucleotide is the building block of nucleic acids. That's what makes that up. Examples of nucleotides are DNA. We have 2 chains of nucleotides formed by a double helix. This gives us our genetic code found in every cell. Another type is RNA. It's a single chain of nucleotides wrapped around itself, and it's made from DNA. And it is what is used. It gives the instructions for making protein. You can remember that from our dinner that we did. Here are examples of your RNA and your DNA. One is single shape, one is double shape. And the difference is the sugar also. We'll talk a lot more about that later. So those are all of our notes. It will give you an idea of what's on the test and what's not on the test. Remember to read your sugar article, know about fructose and why that is such a difficult thing and why it doesn't process the same as glucose. It's important that you kind of understand and know why, if you increase the temperature, that it will increase the rate of diffusion in and out of your cells. How does temperature affect diffusion? Also, understand why the structure of a carbon atom makes it the most important element to living things. That's important, and we talked about that earlier on in the slides. That should help you with the tests. Good luck.